Everyone. Today's topic is culture media and culture methods. Now let us understand what is culture media. Culture media is the food for microorganisms which we need to prepare to grow them in a lab. Okay, so bacteria usually, if we take soil sample, we get mixed population in that sample, and by following appropriate procedures, we can separate them, or we can get isolated colonies on culture media. And this media contain nutrients which supports the growth of the bacteria or other microorganisms. Now this media can be of two types. One is liquid media and the other one is solid media. Now, on the next slide. Medium will show diffused growth, like microorganisms will be evenly spread throughout the medium, and we can't identify the bacteria or other microorganisms based on growth in the liquid medium, because we cannot see the colony morphology or other characteristics in liquid medium, and it is very difficult to isolate in liquid medium especially. And urine or meat broth are used as a liquid medium by Louis Pasteur. And coming to solid medium, on solid medium, we can see distinct colony morphology. We can see the shape and color or size of the colony. So we can identify to some extent the type of microorganism based on the colony morphology. And they are what is colony? Colony is a microscopically visible collection of millions of bacteria originating from a single bacterial cell. It means whenever a single cell is falling on the culture medium separately, it will divide and redivide and redivide, and it will form a group of cells that is called as colony, which is microscopically visible, and it contains millions of cells. And earliest solid medium used was cooked cut potato by Robert Koch. Robert Koch has used potato. He has cut the potato slices and he tried to grow the bacteria on this potato slices, and then he also tried gelatin, but there was a problem with the gelatin as a solid medium, because it was getting liquefied at 24 degrees centigrade, but the temperature required for growth of the pathogens is 37 degrees centigrade, so it could not work out. And then he tried agar agar. Vanni Hesse, um, he he suggested the use of agar in the medium. So it was universally accepted for preparation of solid medium, and it is obtained from a seaweed called as gelidium. This agar agar has no nutritive value, so no need to worry about the addition, or it may how it influence the growth of the bacterium. It has no nutrient value, so it will not affect the growth of the microorganisms, not affected by the growth of the bacteria, and it melts at 98 degrees and sets at 42 degrees centigrade. It means it will become liquid at 98 degrees centigrade and it will become solid at 42 degrees centigrade. Usually 2 grams of agar is employed in solid medium. Whatever media we are preparing, we usually 2% agar will be added in the medium. So coming to the types of culture media, based on their consistency, we are having solid medium, liquid medium, and a semi-solid medium. And based on the constituents or ingredients, medium are of four types. They are simple medium, complex medium, synthetic or defined medium, and special medium. Let us have a look at each type of for this media. And there are also special media, that is under the special media. Examples are enriched media, enrichment media, selective media, indicator media, differential media, sugar media, transport media, and media for biochemical reactions. And the same way, based on the oxidation requirement, we are having aerobic media and anaerobic media. So as we have discussed, solid medium contain usually 2% agar, and we can see the colony morphology, pigmentation, or hemolysis of the microorganisms. And examples are nutrient agar and blood agar. Liquid media, the basic difference between solid and liquid media is agar. Okay, if we add agar to the same media, it will become solid. If we don't add agar, it will become liquid media. Liquid media is used for inoculant preparation or blood culture or continuous culture. Example is nutrient broth. And the third category is semi-solid medium. It consists of 0.5% agar. Example is motility medium. Treponema pallidum motility can be seen in this semi-solid medium. Okay, so here we are seeing three different types of media. This is the solid media. This is the semi-solid media where we can see the motility of the organism into the depth of the tube, and this is the liquid media. Coming to the simple media or basal media. Simple media, it is most commonly used in laboratories. Example, nutrient broth or nutrient agar. It consists of peptone, meat extract, NaCl, and water. So nutrient broth plus if we add 0.5% glucose to nutrient broth, it will be called as glucose broth. If we add 2% agar, it will become nutrient agar. If we reduce agar concentration to 0.2 to 0.5%, it will become semi-solid medium. Now coming to the complex media. Media, what we have seen earlier slide is normal media. Complex media is having more components than the normal media, and here we are adding yeast extract, casein hydrolysate. Here we do not know the exact composition of the components which we are adding. Okay, so that is why it is called as complex media. Then next category is synthetic or defined media. Here main thing is we know exactly what we are adding here, right? We know the exact composition, chemical composition of the medium prepared from pure chemical substances are used here. Exact composition is known. It is used for special studies, for that is example for metabolic requirements. For example is peptone water, 1% peptone plus 0.5% NaCl in water. And next category is enriched media. Enriched means we are adding some nutritious component such as blood, serum, egg, etc. to the basal media. Then it is used to grow bacteria that are exacting to their nutritional needs, which is having special requirements for nutrients. Such bacteria are grown in this enriched media. Example, blood agar and chocolate agar. Next is enrichment media. Enrichment media, it is the media used to isolate pathogens from a mixed culture. Okay, as whatever sample we take, for example soil, if we take, we'll get many types of microorganisms. If we want to isolate a particular kind of bacteria from that mixed population, then we can go for this enrichment media, giving the particular required selective nutrients for that bacteria, we can grow. So this stimulates growth of desired bacterium. Like the nutrient composition will be in such a way that it will support the growth of particular bacteria and inhibit the unwanted bacteria. So it is incorporated with inhibitory substances to suppress unwanted microbes. Example, selenite F broth for isolation of Salmonella Shigella. Tetrathionate broth inhibits coliforms, and alkaline peptone water for Vibrio cholerae. Here there are particular chemicals which are favoring the growth of particular bacteria and stopping the growth of unwanted bacteria. These are the diagrams which are showing different types of media. And coming to the selective media, here inhibitory substances added, and it increases the number of colonies of desired bacterium. Same thing, it is very much similar to enrichment media. Example is deoxycholate citrate medium for dysentery bacillus, McConkey medium for gram-negative bacillus, and so on. And this is the McConkey medium, and this is the thiosulfate citrate bile salt sucrose medium. Next is indicator medium. It contains an indicator which changes its color when bacterium grows in them. Okay, here the medium is giving some indication for the selective organism. Example, if we take Wilson Blair medium, if we need Salmonella typhi, if it grows, if the required organism, that is Salmonella typhi, grows in this Wilson Blair medium, it will give black colonies. Okay, this is due to conversion of some component of the medium into black color. Next is macroid medium. Here potassium tellurite is used. Here diphtheria bacilli can be identified. Next, urease producing bacteria. It will produce urease. This urease will convert urea into carbon dioxide and ammonia. So this ammonia will turn medium into pink color. Okay, so this is the blank. This is after growth. Okay, so after growth, what is happening? The organism is producing urease enzyme. This urease enzyme is diffusing into the medium, and this urease is breaking this urea into carbon dioxide and ammonia is turning the medium into pink. Next, blood agar, it shows three types of hemolysis. Hemolysis means, hemo means blood, lysis means breakdown. It is breaking down the blood cells. They are of three types: alpha hemolysis, beta hemolysis, and gamma hemolysis. This is the alpha hemolysis, and this is the beta hemolysis, and this is the gamma hemolysis. These are three types of uh, lysis of blood cells which we can see on blood agar. Next, differential media. Differential media, here it is used to distinguish between two types of bacteria. It is used to differentiate between different types of microorganisms. That is why differential media. And here McConkey agar or McConkey medium will have peptone, lactose, agar, neutral red, and taurocolide. It distinguishes between lactose fermenters and non-lactose fermenters. The main purpose of McConkey media is to differentiate between lactose fermenting bacteria and non-lactose fermenting bacteria, and this media is consisting of so and so chemicals. So here lactose fermenters will give pink colored colonies, and non-lactose fermenters will give white colored colonies on McConkey agar. Next is sugar media. Here media containing any fermentable sugar. So as the name is suggesting, we are adding some sugar, some type of sugar. Example, glucose, arabinose, lactose, starch, etc. So if a media contains 1% of the sugar in peptone water plus indicator, if we add the Durham streams for the detection of the gas by the bacteria, this type of media is usually used in MPN method, most probable number method, or testing of water sample, where we are adding this peptone water and Durham's tube and sugar to the media. There, these coliforms, if they are present, they will grow and they will produce gas which can be trapped in this Durham's tube. By this gas production, we can identify the coliforms. Here we can see the gas production in this Durham tubes. Next is transport media. This transport media is used for transporting the samples. Okay, means it is not possible every time to take the patient to the diagnostic center, or the diagnostic team can come to the site where sample is to be taken. So sometimes sample is collected in a special media, that is transport media, and it is transported to the diagnostic center, or the place where the testing has to be done. And this transport media is used for delicate organisms which may not survive the time taken for the transporting specimen without a transport media. So this is usually used for delicate organisms. For example, Stewart's medium. It is a non-nutrient soft agar gel containing a reducing agent and a charcoal used for gonococci. Okay, Stewart's medium is used for gonococci, and buffered glycerol saline is used for enteric bacilli. Coming to the anaerobic media, as in the starting we have discussed aerobic and anaerobic media. Aerobic means there is oxygen in that media, and anaerobic means there is no oxygen. Here the main thing is we have to remove the oxygen from the medium, right? They are used for growing of anaerobic microorganisms. Example is robotsums cooked meat medium and thioglycolate medium. Now, till now we have learned the different types of culture media. Now we have to study about different culture methods. They are streak culture, lawn culture, stroke culture, stab culture, pour plate, liquid culture, and anaerobic culture methods. Now, streak culture, it is used for pure culture isolation. Okay, here platinum wire or nichrome wire or loop is used. One loop fill of specimen is taken onto the surface of well dried plate, and it is streaked, or it is moved from one end to the other end of the plate, spread over a small area at the periphery. This is the streaking. First we'll take it here, and we are moving the loop in a zigzag fashion. This is the primary streak. Then we have to go for secondary streak, and then the tertiary and the quaternary streak. If possible, we have to incinerate the loop between the streaks. Then we'll get isolated colonies by tertiary or quaternary streak. This is the these are the isolated colonies: primary, secondary, tertiary, and quaternary streak. Now, lawn culture. Here, lawn culture means growing of microorganisms in a lawn, or more number of microorganisms in a single plate. Okay, so this lawn culture usually is preferred for bacteriophage typing. For isolation of viruses, we use this type of lawn culture, or for isolation of antibiotic sensitive microorganisms. If we want to test any bacteria or yeah, any bacteria which is antibiotic producing organisms we want to isolate, we have to go for this lawn culture. And in the preparation of bacterial antigens and vaccines, for vaccine preparation also we need lawn culture. They are prepared by flooding the surface of the plate with a liquid suspension of the bacterium. As we need more number of colonies on the plate, or lawn culture on the plate, we need more inoculum. So usually more amount of inoculum is poured onto the surface of the plate. So this is antibiotic sensitivity testing. This is the lawn culture. Here we have kept some discs. Okay, so if whatever organism, if this organism is sensitive to this antibiotic, it is showing inhibition, growth of inhibition. So this zone is there. Okay, this is the zone of inhibition. And stroke culture, it is made in tubes containing agar slope or slant. Agar slope, we know if we pour this agar in a slanting condition, it is called as agar slant or agar slope. Here, pure culture, after isolation of pure culture, usually it is stored by keeping it in a slant. And this provides a pure culture for slide agglutination or other diagnostic tests. Next is stab culture. Stab means again we have to pierce into the medium. Here we can see we are going into the medium, solid medium. So here we are puncturing the suitable medium, gelatin or glucose agar, with a long straight charged wire. It is used for demonstration of gelatin liquefaction, oxygen requirement of the bacteria, or maintenance of a stock culture. Here, first thing is we can maintain the stock culture as we are maintaining on a slant. We can also maintain on uh, stab also, and we can check whether the gelatin is liquefying or not by particular microorganism, and we can see whether it is an aerobic or anaerobic by this stab culture. And pour plate culture, here we will take 1 ml of inoculum and add it to the molten agar. Like here we are adding the inoculum into the medium before pouring into the plate. Okay, we have to cool it to 45 degrees centigrade, and then we are going adding the inoculum, and we have to mix it, and then we have to pour it in a plate. And after setting, we can incubate the plates at 37 degrees centigrade, and it gives surface and subsurface colonies. Okay, and with this method we can estimate the viable count of the bacteria. And next, this is the method for pour plate. This first we have to dilute the bacteria, and then we have to pour it, and after that we have to spread this inoculum evenly. And liquid cultures are inoculated by touching with a charged loop, or by adding the inoculum with the pipette. Liquid culture means here we are taking liquid media, and we have to inoculate into liquid media by inoculum, touching the inoculum with a loop, or we can add a liquid inoculum into this liquid culture. Examples are blood culture, sterility tests, etc. So one of the disadvantages, we can't get pure culture over here. This is the blood culture bottles. And coming to the anaerobic culture method, anaerobic bacteria, they differ in their requirement and sensitivity to oxygen. As the name is suggesting, they do not want oxygen. Example is Clostridium tetani. It is a strict anaerobe, and if we give any oxygen, it will die. Okay, grows at an oxygen tension less than 2 mercury. So how to grow this anaerobic culture or Clostridium tetani? By production of vacuum. Normally, oxygen will present everywhere. So if we want to remove oxygen, we have to create vacuum and displacement of oxygen with other gases. If there is a oxygen, we have to replace that oxygen with other gases and some chemicals also. If we use some chemicals, oxygen will react with that chemical, and whatever oxygen is present, it will be depleted. And there are other methods such as biological method or reduction of the dye using chemicals. Next, production of vacuum. We have to incubate the cultures in vacuum desiccators, and displacement of oxygen with other gases is done by using hydrogen, nitrogen, helium, or carbon dioxide. And so here we are ending this topic of using culture media and culturing techniques. We have seen different methods. Thank you.